Good evening and welcome to Do It at DIT, Campus Magazine's weekly dip into the life and times of Dundee Institute of Technology. Tonight, students from Electrical and Electronic Engineering contest the quiz, and Brian Lindsay brings news of the National Union of Students' latest demonstration over the threat to student union funding. But first, our reporter Jimmy Black visited the Department of Molecular Life Sciences to find out about some very advanced research into the molecule from which all known life is constructed, DNA. Everyone nowadays knows that DNA fingerprinting can be used to identify criminals, or even who fathered a particular child. But the technique has many other uses, as Jimmy found out by talking to Dr Karen Sullivan. He began by asking Karen just exactly what a DNA molecule is.
DNA is the molecule which is present in every single one of our cells. It's what we call the genetic material, because it's the molecule which really determines what you're about, what you look like, the fact that you're a human being. It directs everything that happens in your cell, and it's responsible basically for making you, which is why you can use it for identification, because every single one of us is an individual. It's a really fantastic molecule. It actually looks nice. I think most people will probably have seen a picture of the double helix on Tomorrow's World or some of the sciencey programs which crop up on the TV. But over and above just looking that nice, it's actually a really, really active molecule. It doesn't just sit there looking pretty. It's very, very busy at the centre of the cell, really keeping everything maintained and running properly. And if you think about it in terms of things like nerve cells, which don't replace themselves during the course of life, if you live to 100 years old, then the DNA in that cell is literally 100 years old. And every single day of those 100 years, it's been active and working and being used basically non-stop. So it's a very, very stable, durable molecule. Not a lot happens to it. But when things do, that's when problems arise, when it gets damaged, when it picks up mutations. And it's that aspect of DNA which a lot of us are really interested in studying, not just for identification purposes, but obviously a lot of the medical conditions which can arise when DNA gets damaged.
Well, let's look at these forensic police type purposes first.
Okay.
Now, you in a previous life, weren't you? You were involved with forensic science and identifying people through DNA. Tell us about that.
Yes, I was. A few years ago, I worked as the forensic services manager for Selmot Diagnostics, which was the company which were really first to set up the technique for forensic uses. And I was involved mainly then in prosecution work, which is identifying perpetrators of various crimes and saying, well, does the DNA fingerprint, does the sort of barcode pattern that you detect in this person match up with the pattern we detected from the material at the scene of the crime? For example, it could be a blood stain. And it's quite a sensitive technique. You need really just a blood stain about the size of a pinhead to be able to get a nice pattern for identification. So I think this is probably, as you say, where most people will have heard of the technique, because up until the advent of DNA fingerprinting in the late 80s, most forensic evidence was based on blood groups. And as you know, a lot of us share the same blood group, so it's not really a terribly strong way of discriminating between one person and another. Whereas our physical characteristics or our blood groups might be similar, the chances are that our genetic characteristics won't. So it's provided really a big leap forward in forensic identification. It's still not to the level where you can usually say, yes, it's definitely this person, but you can exclude all other people and put quite a high degree of certainty on the fact that if the patterns match, then they would be from the same person.
Okay, but just exactly how safe is it? What's the statistical probability that one person might have the same DNA as another?
Well, it does actually vary in much the same way as our physical characteristics do. Some people have relatively common DNA prints, while others have quite rare ones. The same as, we'll say, blue eye colour or brown eye colour is very common, whereas green eye colour might be quite rare. So the statistics, the probability do actually vary considerably from case to case. If the person who left the blood at the scene of the crime or say had a very rare pattern, you would reach almost certainty. It could be millions or even billions to one that the DNA came from that person, whereas other people might have much more common characteristics, and the statistics that you attach to the certainty of the match might be down to the tens or hundreds of thousands. So there's quite a big range from person to person.
Did you identify any notorious murderers?
Murder cases, not as much. It's more often sexual and violent crime.
Okay, well, let's get away from that now and look at, perhaps for you, the more interesting side of it, because it's more scientifically advanced. You're trying to establish the effect of stress or conditions in the environment on particular DNA cells. Now, tell us about that, and just why are you doing it?
Okay, well, I've been interested in DNA for a long time. My early PhD work and postdoctoral work was involved in DNA structure. And I've really continued that interest, and I'm using DNA fingerprinting to help me continue in my study of what DNA does and how it reacts to different conditions. And another aspect of DNA fingerprinting is that the barcode that you see is really a snapshot of what we call the genome, i.e. the whole amount of DNA in your cells. And what we know is that under conditions of environmental stress, by which we mean increased temperature, overcrowding of cells, exposure to toxic chemicals or to radiation, under those conditions the DNA can change. It alters its characteristics to respond to this challenge. And it's something that we're very keen to understand, because very often the response of the DNA results in a mutation which could lead to, say, uncontrolled growth or tumour formation. So it's something that's important for us to understand. And the technique of fingerprinting is basically a way of scanning the whole amount of DNA to find the small region which is undergoing change in response to stress. So it's really the first time that we've been able to sift through all the DNA so easily to find out what's changing and what's happening when the DNA is exposed to these challenging and dangerous conditions. And particularly now that there's such massive effort and investment going on in breeding and engineering new crops, for example, crops which are disease resistant or can grow in really dry conditions. What happens is people put a huge amount of work into developing these new plants and then find that because their DNA is unstable, these advantageous traits can be lost. Now, we're hoping to find the regions which are unstable and try to understand exactly what's happening, because you can't control these things until you understand them in the first place. The technique is being used as what we call an enabling technology by so many different people, by people working in cancer research to look for mutations in cells, in tumour cells, even to control poaching. People have been taking scraps of little bits of meat left from elephant tusks when they've been recovered from poachers to find out if they were culled from regions where it's legal to keep the numbers down, or whether they were poached from areas of the country where it's actually illegal to take ivory, because the elephants in the different regions have different types of DNA fingerprints. So it's been used to define whether it was poached or whether it was legally culled. So really from everything from medical applications to ecological applications, it's being used by people in all branches of the life sciences. So it's a very, very useful technique.
So the poachers up in the Angus Glens can look forward to seeing you coming after them with your deer stalker on and a test tube and a pipette in your hand instead of a shotgun.
I don't think we got quite to that stage yet, but you never know.
Jimmy Black's report. Coming up after the break is the Do It at DIT quiz and Brian Lindsay's news roundup. We'll be back soon. Welcome back to Do It at DIT, Campus Magazine's dip into the life and times of Dundee Institute of Technology. Brian Lindsay is going to bring us the DIT student news and sport later on, but first, this. And tonight's team are from Electronic and Electrical Engineering. I'd like to welcome Ahmar Ghaffoor, Doug Watson, Donald Laing and Jason Moonie. Now, the quiz is split into 2 rounds. Firstly, it's your specialist subject round, and then we go into the general knowledge round. Now, you've got 2.5 minutes to answer as many questions as possible, starting now. What is Faraday's law of electromagnetic induction? No, blank looks all round. Okay, it was actually the induced EMF is proportional to the rate of change of magnetic flux. In a 3-input NAND gate, one input is logic low. What is the state of output? High or low? Donald?
Low.
It's actually high, I'm afraid. What is Lenz's law? Not to be confused with a law referring to lenses. Any ideas? Nope. Okay, it's actually the induced EMF is of a polarity such as to oppose the change producing it. Now, question 4. How is the bandwidth of an amplifier defined? Donald looks like he's going to answer, and it's Jason.
Minus 3 dB points.
That's right, the interval between the frequencies at which the gain falls by 3 dB, that's right. Why is a long-tailed peer so called? Jason?
2 transistors back to back, and they've got a long tail. The connected emitter.
That sounds about right, because the pair of transmitters have a common emitter resistor. What is the effect of applying negative feedback on the gain and the bandwidth of a simple amplifier? Jason?
Increases gain, reduces bandwidth.
That's right. Does the amplifier in a Weinbridge oscillator have to be inverting or non-inverting? And Jason again.
Non-inverting.
That's correct. How can an analog integrator be constructed from an operational amplifier, a resistor and a capacitor? And that's your last question. How can an analog integrator be constructed from an operational amplifier, a resistor and a capacitor? Any ideas on that one? Donald?
A resistor at the input and a capacitor on the feedback. Look.
I don't know if we'll give you that one or not. Yes, we'll give you that one. Okay, that's right. By connecting the resistor to the inverting input of the amplifier and the capacitor between the output and the inverting input. The non-inverting input should be grounded. Of course it should be. Right, and at the end of that round, that's your first round, your specialist subject round, you've scored 5 points. Okay, so we'll move on now to your general knowledge round. See if you get any more points on that one before we move on. As before, you've got 2.5 minutes, starting now. What name is given in golf to a hole achieved in 3 under par? Donald.
Albatross.
That's right. What is the Monday Club? The Monday Club. No idea? It's the right-wing Tory pressure group. Name the Hunchback of Notre Dame.
Quasimodo.
That was Doug there. Quasimodo, correct. Who played Sybil Fawlty in Fawlty Towers?
Penelope Keith.
What was that, Donald?
Penelope Keith.
No, it was Prunella Scales, actually. According to the nursery rhyme, what time was it when the mouse ran up the clock? Doug?
1.
That's right. The old alliance linked Scotland with what country against England? Jason?
France.
That's right. Which city is Frank Sinatra's kind of town? Jason?
New York.
No, it's Chicago, actually. Who played Jim in Rebel Without a Cause? Donald?
James Dean.
That's right. What sport features strokes, slides and sculls? Doug?
Rowing.
That's right. What is the occupation of a Leary? Doug?
It's lighting the street lights.
That's right, a lamplighter, yeah, going around doing that. When was the National Health Service established? Doug?
44.
48. What was the first James Bond novel? Doug?
Dr. No.
Casino Royale. Name the character played by Catherine Zeta-Jones in The Darling Buds of May. Jason, you look like you were... Any idea? No, it was Marietta Larkin. Who was credited with bringing Christianity to Scotland? Jason?
St Andrew.
No, it was St Columba. The dancer Alice Marks is better known as what?
Groucho.
Afraid not. Dame Alicia Markova. Name one of the states of the USA beginning with the letter D. Ahmad.
Delaware.
That's right. In the film Butch Cassidy and the Sundance Kid, who played the Sundance Kid? Any ideas?
Paul
Donald?
Newman. Paul Newman.
No, it was Robert
Robert
Redford.
Redford.
What is Japan's national sport? Japan's national... Ahmad.
Sumo.
Sumo wrestling, yes. In the book Animal Farm, which animals became dominant? Ahmar?
Pig.
The pigs. That's right, and that is your last question of that round. And at the end of your general knowledge round, you've scored 10 points, which brings your total score tonight to 15. Thanks then to Ahmar, Doug, Jason and Donald, and perhaps we'll see you later on in the series. But now, let's join Brian and Lindsay for this week's DIT News Roundup.
As the NUS campaign against the government proposals on students' unions gathers ground, Douglas Traynor, the Vice President of NUS Scotland, visited the DIT union on Tuesday to put his case. Tomorrow, throughout Scotland, there will be a coordinated day of events called Student Fest, and I asked Douglas exactly what would be happening.
Organised with the colleges all around Scotland, a day of events and entertainments and whatever, really celebrating the best of what student life has to offer from the Student Association point of view. We've organised debates, clubs and societies are rerunning freshers' fairs, we're launching an anti-racism campaign right across the country, and we're also doing entertainments. We've got gigs going on in Glasgow, Edinburgh, we've got discos, one happening here at Dundee University, discos across the country, and we've organised a lot of promotional activity for them, a lot of free gifts and whatever from bars and from various sponsors and whatever. So it looks like being a really exciting event.
I mean, these are the kind of events and the kind of activities which you claim the government measures will prohibit in the future, yes?
That's right. The government proposes to take only 4 activities currently provided by student associations: welfare, sport, catering and internal representation. And everything else basically would go. Now that's everything from community action and charity work and things like that, right through to debates, clubs and societies, campaigns, and indeed the national voice of students, the National Union of Students, would be threatened because student associations wouldn't be allowed to pay affiliation fees to us.
Mm-hmm. Yeah, it could be said though that why should the government subsidise things like student entertainment? I mean, what would you say to that?
Well, the government subsidises various groups in a public way, in the way which it funds us. It funds a police federation to represent police officers. It funds groups like COSLA to represent local authorities. So the representation doesn't seem to be a problem for us to claim that we should be funded.
Tomorrow is a big day for students and staff at the DIT, as it is graduation day. Taking place right through the day at the Caird Hall, there will be a morning session and an afternoon session, and the degrees will be presented by Professor King. During the ceremony, there will be 3 honorary fellowships presented to DIT old boys who have distinguished themselves in their careers. George Simpson, who was here in the 60s and is now a chief executive at Rover Cars and vice chairman of British Aerospace, Alastair McCallum, chief executive at Donenlow Holdings, and Roderick Rennit, the director of water services for Tayside, shall all be receiving their honorary fellowships. And now the sports news. The DIT Football Team is coming on leaps and bounds. After a poor start to the season, they are now 3rd equal in the league after another victory on Saturday against Birkhead. That man Terry Thomas did the damage once more, this time from the penalty spot in a 1-0 victory. Next week they play Fintry Thistle, who are just below them, so keep it up lads, we want to see you at the top of the pile by Christmas. The hockey team had their first game of the season, winning 2-1 against Napier in the Inter-University League. The scorers were Paul Lyall and Robin Campbell. So well done to the boys and girls of the hockey team. The rugby season is about to begin, and I'll have a report of their first game next week. And on that high sporting note, I shall bid you farewell and hand you back to Jeff in the studio.
Thanks, Brian. And that's all from us for tonight. Next week, Jimmy's going to be here in the presenter's chair, and I'll be out and about in DIT. Until then, from me, Jeff Ferguson, it's good night.